Hi, everyone. My name is Julia Apitz, and I am currently a second-year PhD student at Brandeis University in the biochemistry and biophysics department. And I am here today to talk to you a little bit about how I got into science to begin with, and what led me on this path to pursue a PhD at Brandeis. And then I'm going to talk to you guys a little bit about the research that I've been doing in the Kern lab since I joined earlier this year. And so just for some background, I grew up in Methuen, Massachusetts, which is not very far. It's about 40 minutes north of Waltham. And I wish I was one of those people that I could come up here and tell you that I was always interested in science and was always a really good student and really passionate about going to school, but that was not the case. I was always really sick as a kid and missed a lot of school. And as a result, was not always the best student, and school was not really my priority. And I really struggled to kind of, uh, do well in school and kind of find classes that I was passionate about, as well as kind of find this encouragement to keep pursuing or studying and all that. And so I went to high school in Lawrence, Massachusetts, which is the next town over. And in high school, as I'm sure you all had to do, I was required to take, freshman biology. And I actually, really liked it. And for the first time, I was doing really well in my classes and really enjoyed the material and really enjoyed the teachers. And it got me really interested in going to school every day and getting passionate about my classes. And throughout high school, I took a lot of amazing science classes. So in addition to biology, I took chemistry, which is another class I really, really liked, and even anatomy and physiology. And then as an upperclassman in high school, I got to take a lot of AP science courses as well, which I really found interesting. And so this got me interested in going to college and pursuing a degree in the sciences. And I really wanted to go to medical school. So that was one of my big things is I really liked anatomy and physiology, and I was like, I'm going to be a doctor. And so also in high school, I started volunteering at Lawrence General Hospital in the operating room. And so in addition to, bringing patients warm blankets and getting them juice and moving them from surgery to post-op, I also started shadowing, and I would shadow the surgeons and the anesthesiologists. And in particular, I really like shadowing the anesthesiologists and kind of learning about what drugs they were using and their mechanisms inside the body and how they're able to treat pain and different illnesses or infections or, a wide variety of things. And I really enjoyed that experience. And it, was something that really made me want to go to medical school even more. And so I went to college at St. Anselm College in Manchester, New Hampshire, which is also not very far from here. And, I worked really hard in undergrad. I worked a lot of jobs to pay tuition, and I applied for almost every scholarship that I could find. And one of those scholarships just happened to be a first-year undergraduate research experience. And so that was a scholarship I got before I got to college. And basically all I had to do was sit in and take this first year undergraduate research experience course. And then I did lab work over the summer. That was part of it. And in return, I got this scholarship. And so I thought, great, this is a great resume booster. I'm going to get lab experience. And I get a scholarship out of it, which is awesome. It's a win-win. And so the summer that I first did research, I did research in an organic chemistry lab. And I was looking at green methods of aromatic nitration. So basically how to synthesize these compounds in a greener method that creates less waste. And I actually really enjoyed the lab research, and I really enjoyed kind of the experience of getting to figure out this problem and getting to solve it using scientific research. And the next summer, I even moved labs to a kind of a more biochemistry lab. And I also switched, I entered college as a biology major, and then I then switched to a biochemistry major because I got to take an even wider variety of courses like chemistry and physics instead of just kind of being focused on biology. And in my biochemistry lab that I ultimately did my undergraduate thesis in, I did work on discovering small molecule inhibitors of the Hippo pathway. And so the Hippo pathway is important because it is involved in a lot of different cancers when dysregulated or mutated. And so finding small molecule inhibitors was really quite interesting. And so I have a couple pictures here from different conferences. So in the top right, I have the first conference I ever went to, which was after my first summer of research, and my dad was there and got to see me present at my first poster presentation. And then in the lower left there, I have, I think this was before my senior year of college, and this is me presenting my work on the Hippo pathway. And then in the bottom right, I have me presenting over Zoom during COVID at a conference. And so it was really cool getting to go to a lot of different conferences and get to describe my work to people and get really enthusiastic about it and share it with everyone. And so it was at this point that I kind of started to think, maybe I don't want to go to med school and maybe I want to go to graduate school instead. And a lot of my professors would talk to me about it. And graduate school was something that was not on my radar at all in high school. And even when I started college, a PhD was not in my cards at all. And so I then started to look at PhD programs as a way to kind of like continue to do this lab work and this research that I was becoming so passionate about, and then eventually use this later on to continue my research and kind of find ways to help people and to look at different diseases and how they can be treated. And so my senior year of college in the fall, I applied to graduate programs. And I also applied to a lot of internship programs, and I got an internship at Biogen for my last semester of college. And so at Biogen, I was in the biologics drug discovery group. And so they're the group at Biogen that had, produced the Alzheimer's drug that has been in the news a lot. And so I got to, do a little work on that and kind of see how a lot of the scientists there that had pursued PhDs actually were kind of the people in charge of that project and had kind of seen it from its inception into getting released. So it was really cool to see that. And it only further cemented that I wanted to go to graduate school and get a PhD and do this kind of work that I was seeing people do in biotech. And so I got into Brandeis University, which was my top choice. And I started in the fall of 2022 and did my rotations my first year. And then I joined the Kern Lab in 2023, so this May. And now I'm going to talk to you guys a little bit about what I've been up to in the Kern Lab since joining. And so my project focuses on the circadian rhythm. And I'm sure a lot of you know about the circadian rhythm and kind of its role in how we respond to things throughout the day and even at night, and kind of how your body is able to prepare for like its rest period at night and is able to keep you alert during the day. And so I think this figure here is kind of a great outline of, what's going on in your body throughout the day and how that's regulated by the circadian rhythm. And so, like at nighttime, you kind of have down here, like where your body is preparing for rest, your body temperature rises, you start to secrete melatonin. And then at 2 a.m., you kind of hit your deepest sleep. And then what I think is kind of interesting here is like you get your high alertness starting at 10 a.m. So while you guys are all in class, and then kind of after school at 2:30 or 3:30, you have your best coordination, your fastest reaction time. So like when you guys are doing sports after school, that's your body having its best reaction time and best coordination. And so the circadian rhythm is really important in regulating a lot of different things. And so while we know when we have, highest body temperature, best coordination, not a lot is known about the mechanisms within your body that are actually regulating this and, keeping this 24-hour clock in place. And so the question I kind of want to answer is, how is this 24-hour clock maintained? And so Michael Rosbash is a professor at Brandeis University, and he actually won the Nobel Prize in 2017 for his discovery of molecular mechanisms controlling the circadian rhythm. His lab works with the circadian rhythm of fruit flies. And you might not have thought this, I certainly didn't think this, but some of the proteins that are involved in maintaining the circadian rhythm of fruit flies are actually very highly conserved to the proteins in humans. So you kind of have something in common with fruit flies, which is funny to think about. And so in humans, the core protein that's involved in maintaining the circadian rhythm is casein kinase 1, which helps to maintain kind of this core clock. And so what is a kinase? So a kinase is an enzyme that catalyzes the transfer of a phosphate group from ATP to its substrate, which I have highlighted in this figure down below. And so this is actually kind of a fitting figure because proteins are made up of amino acids. Those are their building blocks. And so the amino acid that I'm looking at is serine. And so this OH group here is very representative of the serine. This is what the R group of a serine looks like. And so once this gets phosphorylated, you replace that OH with the phosphate group here. And so that's what casein kinase does, in particular to its substrate, which is human period 2. And so I have H-per outlined in the figure on the top in pink here. And so this is H-per in pink binding to CK1 in green here. And then in the little spheres, kind of on the right, I have the serine residues that get phosphorylated by CK1. And so this is kind of the structure of CK1 and H-per, which is, something really cool that we can visualize and kind of see how proteins interact with each other. And so my project specifically wants to look at this interaction between CK1 and the PER protein. And so on the right here, I kind of have a diagram of roughly what's happening in the cell with CK1 and H-per. And so in the cytoplasm here, you can see that H-per gets phosphorylated by CK1, which then binds to this other protein called Cry, and they form this complex and are able to move into the nucleus. And so within the nucleus, they block transcription of period and Cry. And so during the late night, early morning, you have high levels of this complex in the nucleus inhibiting transcription. And then in the late day, early night, you kind of have high levels of these free proteins in the cytoplasm. And so, this is something that could be dysregulated, but it's not really known what their effect might be on keeping this 24-hour clock. And so there are also several mutations that have been found to alter the circadian period. They've either been discovered in fruit flies or in humans. So it doesn't matter if they've been discovered in fruit flies because, as I mentioned, they're conserved in humans as well. And so there's also another mutation that was discovered in PER. And this was first discovered from a large family in Utah. And so they have this single amino acid residue mutation. And this shortens their circadian period from 24 hours down to 20 hours, and in some cases, even 18 hours. So a really significant change. And as you can imagine, really throws off, how they're able to function during the day and night. And so this kind of highlights the importance of what the circadian rhythm can do. And dysregulations in the circadian rhythm can lead to a lot of problems. So not just, trouble sleeping, but it can also lead to weight gain and depression. And it's also been connected to a lot of diseases like cancer and Alzheimer's and cardiovascular disorders. And so there are a lot of these mutations that have been discovered, and we know how they alter the period in terms of, either shortening the time or lengthening the time, but the mechanism by which they alter the clock is not known. And so from determining the mechanism by which they alter the period, we can learn a lot about how CK1 and PER interact in order to maintain the 24-hour clock. And further down the line, it might be of interest to see if, these people with the mutations can be treated, or ways that you can get your circadian rhythm back to a healthy level and not have this dysregulation that can lead to a lot of different health problems. And so here I have on the bottom right a lab picture, and I just have, a quick message for everyone that you can achieve anything you set your mind to. I was not always into science at all, and once I got interested in it, I really worked hard and became a better student, and really worked to kind of achieve this goal that I wanted to of pursuing my passion for science. And there are also so many amazing paths in science and areas of research. I was not, always aware that graduate school was an option that I'd be interested in. I always thought I'd wanted to go to med school. And there are just so many great areas of research to look into, whether it's biology or chemistry or physics. The possibilities are almost endless of what you can do. And my last piece of advice would be research and internship experiences, whether the internship is in, a hospital or a biotech or even a lab, an academic lab. These experiences that you have, either you can even do it in high school, in college, and beyond, are such a great way to find a path that you really want to pursue and are passionate about. And so with that, I'd like to thank you guys for listening to my talk.